Right. So let me know if you see that screen quite well.
Yep.
Yeah.
Okay, good. All right. So thank you for the opportunity to talk about the challenges and opportunities for trying to automate some aspects of metadata and histology and microscopy. So my preface here is that this is a very old art, but because you can store those blocks for years, you can get access to archival samples. And I'm mostly going to be talking about formalin fixed and paraffin embedded sample preparation. But I think a lot of those things that we've done are applicable to other microscopy methods as well. And even a couple of years ago, you couldn't really do much with those formalin fixed paraffin embedded samples, but that changed quite a bit. That now you can do high-plex immunofluorescence, you can do spatial transcriptomics assay, and similar things. So we make a lot of slides and a lot of blocks, and efficiency is very important for us. We cannot really spend too much time on it. We provide help to a lot of labs and users, and our billing is really complicated. So my personal journey into histology was that I was bad at it. I was really, really bad. I think I made all of the mistakes that were possible to make. The old rule of garbage in, garbage out. Thankfully, I had been working with very talented histotechnologist who held my hand, and she essentially teach me how to do it right. One of my biggest problems was that I did not have a system for my record keeping. So an enormous amount of time was wasted because I was essentially going back and forth, but I was persevering in my badness, and that was motivating enough to develop a system. The problem was that I was changing that system, and that created even more mess. So those are sample IDs from my single experiment. As you can see, there is quite a bit of evolution here. So in terms of just very broadly and generally, from a perspective of a core lab, we definitely need to know who is the owner of the sample, and if that sample is safe for us to handle, right, at the minimum. Then the thing is that the slides and samples must match. So we will need to make sure that the slide that's been cut from a tissue block matches it. It needs to be done for every single item that we make. As you can see, handwritten labels are not the best. More than that, slides often need to be cut in a specific order. It would be ideal if the slides that we digitize would inherit all of those data that were accumulated along the way, and data should be fair, right? So if you will create a system that allows your users to find their records, access them, reuse them, and ideally this would be interoperable as well, you can get the best buy-in. And that is a very important, probably the most important statement in my presentation, is that every person that was going to be using this, either user, core staff, or PI, needs to see the value of that system. And storytelling helps. So my story is that imagine that that you're writing a proposal, and you know that 5 years ago there was a study that was done where they used exactly the right genotype of mice and exactly the right treatment, and instead of now frantically trying to repeat that experiment, you'd be able just to take more sections from those blocks, or even reuse the slides that they've done and analyze your aspect of their phenotype. And I'm telling them that, you know, this person could be you, or it could be someone who is coming 5 years down the line. So we can help you to get better at organizing this, and everybody is going to be happier. Now, people are not happy when they use Sharpie to write on the cassettes, because that does not survive processing. But there's sometimes some remnants of fluorescence that you can image with a whole body imager like the AIMM HD. So this is a video that shows the workflow that's typical. We start with printing histological cassettes. They're given to users. They bring their samples in those cassettes. We embed individual pieces of tissue in paraffin wax after processing. They are being sectioned. And I love this moment of the video. I hope it's not playing too choppy on your end. Yeah, there is a lot of art in this kind of work. And we try to make sure that people don't spend a lot of time like hand reading those labels, you know, and trying to decipher if it's L or I. A little bit of time saving goes really a long way. Those are our machines that automate staining and cover slipping. It would be awesome if we had a machine that would cover slip immunofluorescence slides, but that doesn't seem to exist. And then everything gets, almost everything that we do gets scanned on our AxioScan slide scanner. It has a barcode reader. And you know, it took us maybe 7 years to come up with a kind of a solution that would satisfy our needs. And before I go into that, I want to tell you that you can start your labeling process on a budget that's less than $100. And this Brother P-Touch printer allows you to print on tapes that are surviving tissue processing, and that you can also put on your slides or any other type of material that you're generating. So this was version 0.1. I didn't count the versions. This is just a joke, but there were many versions in between. So speaking a little bit about our data structure, all items must have a unique identifier, okay? Everything needs to be differentiable. The work information flows through the work orders into the samples and tissue blocks and slides, and those slides get stained. So there is certain parent-child relationship as the slides inherit the IDs of the tissue blocks. It was very important for us to make sure that the human data are human-friendly data are very prominently displayed, so it's easy to read that, and that computer-friendly data are organized in such a way that they allow the system to keep running. And we also wanted to build some redundancy in case the barcode will fail. So this is how it looks like on the histological cassette. We have the barcode, and we have the information about the work order, the number of the cassette. This is something that we assign. The sample ID comes from the end user as well as the organs. We adopted this, we essentially don't enforce specific sample IDs. It's up to the user, but we suggest that combination of letters and numbers is the best. And obviously, human samples must be de-identified, something that's super important. We highly recommend not to use mouse 1, mouse 2, mouse 3, because in the next experiment, you're probably also going to start with mouse 1. In terms of the slides, a lot of this information is inherited. So we have the work order ID. That's our Kanban. That's our group of blocks, slides, and derivative materials or scans. We have our sample ID. That's mostly for the user. We have the organs or other kind of information that we want to put on the label. And we also put email address of the person who requested it. It's easier for us to essentially look at the slide, know who it belongs to. There's our barcode for the slides. It's slightly more complex than the one for the cassettes. And there is cassette ID and a slide number and stain. So the way how it's implemented for the end user is that we use our core management software that's called Infinity. And what I wanted to highlight on this slide is that you really need to work with the user and educate them how to use that system. Unfortunately, these things get complicated. You kind of need to ask a lot of questions. You will see them in a moment in order to gather all of the necessary information. So a few people read those instructions. A few more people watch my step-by-step explanation. And I highlighted the first video that kind of paints this rosy picture of how good your sample processing will be if you follow those easy 77.5 steps. That's essentially selling them the value proposition of this whole thing. And one of the ways how I encourage them to do that is that your materials and methods will be recorded. Essentially, the most critical component of what you need to know will be recorded through that work order. We need to know those things, and you need to know those things. And I'm putting a smiley face next to the BSL level because I actually made a mistake. MCNB as BSL1. And there is a bunch of things that are histology specific that we ask for. There is even more of that. We can receive those work orders, and we can actually pre-print cassettes for them. So they get those cassettes with the barcodes, with clear labeling, and they take that to their places where to generate the samples, and they can put the right organ into the right place. They don't have to, scribble something on a jar of formalin and stuff like that. If they need slide scanning or immunostaining, they also make those notes. But for the slide scanning, that would be the only thing that they need to fill out. But if they need an immunostaining, that's another form that's quite elaborate again, that collects the data that are necessary. So the most important part from our perspective is actually the samples. With the web form, you can enforce certain way how those samples are entered. So for example, we only allow 8 characters for the sample ID because more will not fit on the cassette. There is also short description that will get onto the slide. There is information about the organ, and we make it mandatory because certain organs need to be processed a certain way. Experiment information, that's for them to write as much or as little as they want. We need to know about how many slides they want, what kind of slides, what kind of special stains they may want to have. And in this form, we built in a way to link previous work orders. So they can put in the 4 digits that are on each cassette. So we can kind of piggyback on the information that was already in. And this is essentially reserved for future development. If we want to add something and not change all of that layout, we can kind of put it in here. So the very painful but necessary element here is the billing. This is how we can add on supplies, itemized charges for our work, and people are selecting grant, account, purchase, order information for invoicing at the very bottom. Okay, so at least in theory, after the order is submitted, all of that information is easily accessible, both by the user and by the core staff. And we can edit the stuff. So I fixed that BSL1. We can add, delete, and modify samples, and that is very common. People bring more stuff, or they forget something, or something didn't work, and they have 3 samples instead of 7. And we can export data to Excel but we can only export this table. Unfortunately, nothing from this is easily exportable in that system. So there are quite a bit of limitations. That's also why this form looks this way, because we can then take that data and with help of a bunch of weird macros in Excel, we can add and transform the data to output information that's going to be used to print cassettes, slides, and provide information about the stains, and it has a bunch of other functions. So I'll leave you the history of how it got developed. In 2019, we rolled out this core management system. In 2020, COVID was absolutely decimating and killing our workflow because up to this point, we relied on in-person communication. There was lots of notes on the paper. When that in-person communication was gone, so was our workflow. So mid-2020, we tried to move this whole workflow to Infinity, so we have one place to go online and check where the samples are. And at the end of 2020, two things happened. We got internal grant money to fund more histology automation, and that focus was on data handling. So we had more computers, barcode scanners, barcode printers, and a bunch of other hardware. And our hero developed a temporary workaround to our label printing problems. I would like to share with you those 3 lessons for wannabe developers. You're going to be trying a lot of stuff until it works. Temporary workarounds are not a thing. If you put something in place, it's going to stay. But I would also like to try to encourage you not to try to think through every single thing. It is impossible to predict everything from the starting point. Some decisions need to be made and you need to stick to them. Obviously, designing that database is tricky. And if you do some bad decisions early on, there's going to be pain, but there's probably going to be pain anyway. So just go ahead with it and give it a try. And this is our hero, Sarah McCardle. She's a senior staff scientist. And she's a Chan Zuckerberg imaging scientist. I think this funding actually helped us to kind of, open up the heads a little bit and say, okay, she can essentially not do the work that we would normally charge for, and she can focus on giving us some stuff for the future. So she is the first person to say that this is not the most elegant piece of code. However, it runs. So this is how it kind of works on the staff core side in terms of getting nice labels printed. All of the files live together on a shared Google Drive, and we have the Google Drive file stream installed on all of the computers, so we can sync the data in between. The nice label printing software is our favorite one. That gets connected to the Excel database. From there, or from that database, actually, we can send data to cassette printer. There is that part does not involve nice label. But then we use Dymo printers for slide boxes, cassette boxes, things like that. Those labels are very cheap, and you can make them in many different shapes. Zebra printers are something that we begin for slide labels, and later on we got a slide printer that can print directly on the slide. So if you would put slide with a label on a confocal, and you wouldn't have anything on the other side of that slide, that would actually cause some tilt. So although those slide printers are quite costly, they're the best possible way to go. Now, after the scans are done, each file name is basically this barcode plus the date of scan. So Sarah wrote this piece of software that is essentially looking within the metadata of Zeiss AxioScan images, and it extracts the duration in milliseconds for how long did it take to scan the slide. And that gets summed up, and this is what we're inputting manually into the Infinity for billing. Because we have this connected also to the database, we can do some custom renaming, and this script you can find on Sarah's GitHub page, where now each this barcode is very short. It's as small as possible, and there's also some QR error correction enabled here. But that wouldn't be too useful for the end user. A bunch of numbers are not as good. So we add work order ID, the email of the person who needs to get the files, cassette ID, sample ID, short description, the sequential number of the slide cut from the block, the stain, and the date of scanning. And this workflow also supports IF optimization slides that we're doing quite a bit that have different needs for labeling, and IF multiplex up to 6 channels. Now, there's a gazillion of things that still needs to be solved. Our biggest problem that we cannot actually manipulate the metadata in the CZI files. At least we haven't found a way to do that. So you can go and manually type something into the software, but you cannot code a script that's going to do it for you. So we definitely want more streamlined implementation. It's a temporary workaround, as I mentioned, that stuck and works very well. We want more automation in generation of work orders. It takes a long time to put that information together. And we crave for more automated billing. What we need is better support for multiplex slides. So now we have a system that can stain 17 or 20 channels. We haven't figured this out. We didn't even start. There's this minimum information for multiplex tissue images. And what we do now is essentially we say, okay, this is panel A1, this is panel B2, and we save that. But there is a lot more that these good people from MITI Consortium are asking for, and I have no idea. We want more on automated image data archiving. We have Omero instance, but things are not connected yet. And we need somehow some way of transferring the metadata to Omero. So with this, I would like to thank you very much. The people on the left are part of our team. They're all brilliant and lovely. I would also like to thank Pete Bankett and Mike Nelson, because next week we are organizing CUPA training course at my place. And if you want to join online, you can still register and
okay.
send you a Zoom link. All right, so thank you very much, and thanks for the Chan Zuckerberg Initiative for funding.
Thank you so much. That was amazing. And I think we should go directly to Ken, and then we're going to have questions and discussion afterwards on both of them. That's okay, Judith? Okay.
Okay, then I'll share my presentations. Sorry, I think my presentation probably not as good as the previous one, nor as probably as useful. But I think we kind of hopefully that we can help. We can get more lesson from what we encountered, and see what some of you may have actually solutions to help us on that. So our work is trying to systematically annotation of images. This pretty much a work in progress situations. Because I'm actually from the UK, so I just quickly introduce the Francis Crick Institute. Is an amalgamation of 2 institutes before. We have about 2,000 staff, about 1,200 scientists with 120 labs. More importantly, we have a very inclusive and supports diversity. So what happened is we like users to use whatever methods they want to use. We don't dictate to them what they can use and not to use. And so it also comes with a lot of problems when we try to annotate data in our process. So I'm actually part of the advanced light microscopy, one of the core facilities. We have other core facilities, including the EHP and other facilities like AIMM and things like that. So I was mainly on the light microscopies. We have something like 35 large systems, about 20 others, total about 55 systems. You can find some of our systems here on equipment, things like that. So you can find all sorts of different systems that we actually run on. In particular, I mentioned about some of the slide scanners. We also have the size actual scanners at the EHP facilities. We mainly provide a lot of around 75% of our group actually use our facilities. We do a lot of training per year, and the user uses our systems to acquire the data. We actually run about our Omaro for about 2 years now. We have about 130 terabyte of data there, about 280 registered users. So you're talking about a quarter of our staff or scientists, only a quarter of it, and about half of the groups actually kind of know Omaro or use Omaro, etc. But there are some active users, about 17 users kind of last month, just a example of importing about 4,000 images in the last month. Last time, I think we mentioned about Omaro. It's very difficult to get users to actually use it, and it is true. Actually, we have the same problem, but we find that there is two main interest for people who want to get people to use Omaro. One is this slides histopathologies. The other one is to use AI pipelines. So we have a pipeline that we use slides, and we store the slides and annotations in Omaro, and then the machine learning team will then have a workflow that will grab all those data and then try to analyze the genomic data, annotate it with the images, and from the images get some results and things like that. So that is there's a few projects based on Omaro, using Omaro with histopathology slides and doing machine learning. At the same time, the other part that is got used quite a lot is about our labs that have external collaborators. Instead of sending images across and things like that, we find that they can actually use Omaro. We have an external access, so they can actually share annotations, ROI, and discussions on Omaro using it. So these are the two main applications that people use Omaro for, and they are quite active using that. The problem, metadata. We have a lot of for some projects, we have very detailed annotations of genomic data attached to each image in some of the projects, but they're very project-specific labels, and most of the image, however, have no metadata attached to it. So what do we want to do? What kind of metadata do we want? For us, our objective is trying to understand what the contents of the images are. So it enable better searching, accessing the image data of certain type. We can reuse them for data mining and machine learning. We can also compare in this sample and images from different microscopes because we got different microscopes, so we can actually, see which microscope provides better solution, etc. And by doing that, we are mainly actually targeting the RAMB biosample metadata. So it's slightly different from what we normally do, is to look at core kind of microscopy images. So what we are interested in, what the images are, what the contents are. So mainly we're interested in looking at the organism, what we are looking at, and how do you what can it show, what kind of dye it use, or what kind of stain it use, etc. And with that, so we spent a few hours on getting together all our colleagues, trying to come up with a metadata for biosamples based on RAMB. So the black label is the RAMB one. The blue one is the one that we think we want to have, and this one of those kind of examples that we think will be what we want to annotate, what we want to get out of, and we want our users to annotate them like that, such that when we have an image, we can tell what we're looking at, what type of dye they use, what type of fluorescent they use, etc., what they're looking at, etc. And however, we tried out on two different type of data that we actually collect. One of them is that because we located in central London, and we have and we buy a lot of equipment too. So the microscope vendors do a lot of demonstrations of their latest tech at our facilities, and a lot of our users then actually use those microscope and then generate the data and then check them out and things like that. So we collect a lot of those data, and we think that this is a good way of using those data to check out whether our metadata format will work. The other part of it is our existing slide scanner. We actually have two of our slide scanners actually is connecting to the network storage, so they write directly to the network storage, and then from there we have a semi-automated way to transfer and import those images directly to Omero. So we thought that we can actually add to that the metadata. If we ask the users to supply the metadata, we can import them together with their images. So that was the assumption, and that was the thing that we tried it out. In reality, we found that there's a lot of problem with that. One is that each image file may actually contain more than one images, and they have different metadata associated with them. And so we found that we can't just import the metadata together with the image together. So what we can do is to post-process the data. So we have to import the images, and then we create a Omero script that will output the spreadsheets to users on all the images, such that we can match the annotation to those things. But however, we find out that some of the modality do not fit into our metadata. For example, we have phase contrast images and things like that that would not fit this idea of having channel 1 fluorescence, channel 1 molecule of interest, channel 2 fluorescent, etc. Some of them do work quite well, but some of them, for example, here we have got basically got 32 spectral channel, and so we can't really fit things in correctly. That is mainly to do this. The latest one is actually using the Nikon, the latest Nikon microscopes with the resonance scanner, and we are trying it out and trying to label them. The other thing is about tissues. Earlier on, we had the speaker was talking about labels. We do have labels and of the images, but here what we have is 3 different tissues, and they are different things, and it is in one slide. So our workflow or our assumption on just having one data set bit difficult on implementing it. So here is actually a user supplied metadata. So that is the metadata that we're receiving. So it's a bit cryptic, but I mean, some of you are expert in biology and probably know what this is all about. Extracting it separately, what we can tell is that basically on one of the slides, we have animal 1 with a genotype, so wild type, kind of mouse strain, and then on the other one is on the same slide with a different animal with a different genotype. So that becomes obviously that it is not easy to basically annotate data based on based on what for example, based on this slide, which have 3 different tissues. One is control, whereas the other 2 is the main tissues, and how to explain it in a way that systematically we can utilize it for example, machine learning or accessing it and things like that in the future. So summary, we find that it's not so trivial to use a single metadata format to capture only a subset of microscopy output. We may need to develop multiple metadata formats to fit in different things, but then it becomes a problem of implementing it, the problem of training users to use that, etc. So it's still work in progress, and hopefully some of you can give us some suggestion and comments. Very welcome on this. Thank you.
Thank you so much, Ken. This is, I mean, Judith and I are smiling because we are super happy about both presentations. This is exactly what we were hoping, so that we can actually exchange information about multiple things. And actually, it's interesting because teams are emerging. And that's what I was also hoping when we started out with this weird idea of having this user group, that there might be, you know, but going through different experiences, we might end up with some themes that are shared and that we can then maybe start working on in, you know, subgroups or whatever. One theme that I'm recognizing is first of all, amazing stuff that Z did about the barcoding, you know, we haven't even started thinking about that. So that's all great to know that, you know, if we start thinking about it, then we can ask somebody. The other thing is this, you know, obviously automated import in whatever platform. It seems like even Z is interested in Homero, or at least exploring it. That's another big theme. And the other big theme is how do we cover the variety, the complexity of the different experiments in a way that makes sense, doesn't try to get like an orange in a square box, while at the same time not leaving it something that can be handled. And I mean, I'm just going to say what we have been thinking, but then I want to open the floor. Is the idea of using REMBI, expanding it like you did, because basically you took some fields from REMBI specimen and then added more, which is the same thing we have been doing. So that's I mean, that's validating the kind of things we've been thinking. And the other thing is modular REMB extension template. I don't know what to say. So having a sample, biosample, and then different maybe modules of metadata fields that can be swapped in and out for the sample, for the specimen, and for all of this stuff, so that we can start sharing it. So maybe in one of our next, maybe in December, we can start thinking about how that could, maybe that would be, we don't have a speaker necessarily for December. So one possibility that I'm just now brainstorming is that we may want to meet and start thinking about, for example, how would we share these possible templates. That's a suggestion, because I think it's urgent that we start having, we put up what we have and Ken puts up what they have, and we start comparing notes. Anyway, this is what I can see as so far themes that have emerged. Judith, or any other person that wants to comment?
Yes, so sorry, I was, does anybody, everybody knows what REMB is, or maybe not? Raise your hand. So essentially I was typing.
Okay. Yeah, so
I was
I'll put
typing
it.
this. So you put the DOI if you have it.
Yeah, I will
Katrina.
put it there
Okay,
while you're talking.
thanks.
Okay.
Yes, so
sorry, thank you for that.
So yeah, that was really amazing. So and as Katrina says, we're really grateful to that you guys are sharing your experiences with us. So we are, thank you, Christian. Christian is author, actually, so we have the honor of having Christian presenting. So it seems to me that today's theme is how do we organize the experimental design and sample preparation metadata. We've been using a spreadsheet, Ken as well. The question I have for you is your web form. How can you provide us more details on how you got organized with the web form? And does it imply a spreadsheet at one point or not?
Yes. So the web form is there because we needed a way to tie it to billing. That was the important thing. We started with spreadsheets that people were sending in, but the problem was that they were very liberal about the things that they were putting in, using their own columns, putting 20 characters in a field that needs to have 8 or less. So in certain critical parts, a certain constraint of what the user can do is actually pretty good, because we are physically limited by the amount of space. From that perspective, right? But I would not necessarily recommend you any core management system. I think they are all pretty bad. And I always have a hate and love relationship with them. We kind of have it because of that billing issue. But this could be done in a much better, much easier way with modern web tools. And I also wanted to kind of point your attention to the fact that this natural language processing that bloomed over the Metagram, I believe it's called, and ChatGPT. There are things. There's an app on my cell phone called AudioPen, where I can ramble for 15 minutes, and it's gonna translate that. Even with my weird accent, it's getting a pretty good translation, and then it sends that to ChatGPT that shortens this or transforms my text into something that's either humorous, or it's a pointed list, or it's something else, right? So there is this I think it's a startup. I learned about them a couple of days ago, haven't checked them out, but apparently they essentially place an Alexa in your lab, and where you can connect a bunch of things. But the major point is that you talk to it. You know, when you're doing the experiment, you can actually talk to it, and it will record the stuff that you're saying. And having seen what those systems can do, I think it will be essentially in a pretty short time possible for us to just record as we're taking microscopy images, and there's going to be enough structure in that data that the system will be able to automatically figure out that this is the staining, and that's the sample ID, and that's an artifact that I saw, and that area needs to be avoided. The other thing that might be related to Ken problem is that image recognition got pretty far, and we haven't played with this, but it seems to be doable to essentially have a neural network that's going to recognize those images and classify them. Your control is quite stereotypically placed in one end of the slide. So you can also kind of build in some rules on how to separate that. Now, those things do not exist, but I remember a couple of years ago, we were planning on doing something like this lab voice AI with one of the postdocs here, and we didn't know what we're doing, so it never got anywhere. But you know, things that we envisioned 2-3 years ago now are realized in practice. So I think that that's going to be the thing that's going to allow us to wrangle that massive amount of very various metadata.
Yeah, I think I'm just wondering whether, I mean, there's another debate that we have been internally was saying that, you know, do we need to have metadata format, or do we need to have this metadata thing? Why don't we just let the users have a free text, and then we use like ChatGPT or whatever search engines that will, as what C was saying, you know, you do all the searching, then you actually can figure out the data. I think my thing is the other way around that we are looking at the moment is, can we annotate them such that the system can learn? So I think it's the first part at the moment. We are trying to say, hey, we've got a lot of image data, and we've got a lot of internal knowledge about those images, but we haven't been able to put it in a way that the machine can learn. Can we help them to do that? And then later on, we don't need to do all these things, because as what you said, the system may be able, whatever image you scan, they already tell you what it is and what things to find and what you should look at and things like that. But I think we are not there yet, I think.
Is there anybody who have a comment or question before I say something? Unless somebody else.
I think this is good. This is good.
No, I just want to be sure that other people, that I'm not taking time from other people that maybe want to speak.
I'm not.
Okay.
Go ahead.
So this is very interesting. I mean, of course, we have to make sure that users are, you know, that things are done as easily as possible. And I think language recognition is definitely going to be part of that picture. I am pretty sure. What I am not convinced about is, I don't think that, you know, if the problem, so there is two aspects. One is the usability of the system. And so for that, I think that is, I'm all in. What I am a little bit concerned about is thinking that that will save time in terms of then comparing the results. So if we're saying, you know, one thing is for the user to find their own data. And so what I call pre-publication data management or pre-publication stewardship, that obviously they are the only one that needs to understand it. So that's totally fine. But if we then need to share, which is definitely increasingly becoming important, we still need to have some way of communicating with each other. So I'm just wondering, do you think, I mean, I think you have looked into it more than I did for sure. So, for example, if we come up with, for example, something like a shared set of templates for different aspects of the metadata, and we agree on certain things, do you think that those systems can then fill in that structure? So can they use the language to actually map to for example, fields, so that at the end ends up being something that the machine can match with can be used to find and other things? I mean, it sounds like this is probably going to be possible, but I'm just asking what you think.
It does, but those language models are statistical, right? So if they have not heard enough, or they didn't see enough on the internet about certain things, they will be making mistakes and they will be hallucinating and confusing things. But they become much better, and I think there might need to be certain branch of those systems that will be specific for labs, that kind of learned all of that metadata already, and they kind of recognize that structure. But in terms of just being able to, chop your rambling into individual tokens and like, aha, I think it goes here and I think it goes here, that's probably doable already. And I think then, having user in the loop, who instead of having to type it, will look at the spreadsheet and say, okay, this is wrong, this is wrong, this is good, everything else checks out, would be a major improvement, right? I mean, if lab works with mice, and they never use zebrafish, there are also some things that can be implied and things like that.
Mm-hmm. Yeah, no, definitely, it's a good idea. I mean, it's I don't think it's gonna allow us to cut the corner, to come together to decide what information we need. So I think that we still will that hard work of harmonizing. I'm not going to say standardizing, because I think we're not, for sample preparation, and it's going to be very hard and maybe not even possible. But I think harmonizing, as wherever is possible, it might be a good word. I think that kind of work we still will need to do. But I think everything that we need to do, we could do to make it then actually, I mean, doable for the users, then obviously we need to do. Okay, we actually have only 3 minutes, and we don't have a speaker we have a speaker lined up for February. It's going to be Stephanie and Susanne from NFDI for Bioimage. Are going to tell us all about the amazing stuff they're doing. For December, is there anybody here volunteering to present their system? Somebody that have already their stuff, they're already doing stuff, or other people that are have challenges that they don't know how to solve? Christian.
So I'm speaking on behalf of my colleague Tom, who had to leave the meeting earlier because he's currently at a conference. And Tom has done a lot of thinking into the structuring of the tagging and the data organization in Omero to actually, so to say, lessen the necessity for automated uploads by thinking about how the data will be structured in the end. That's also based on Rambi, and that is done in the framework of the i3D Bio project. You could call it the little sibling project of the NFDI if you want. It's basically a little workforce that came from the RDM for MIG group, which you have heard of, which is, so to say, sort of a parallel group to this in Germany. And we could, I think, invite Tom for next time. He volunteered.
Great.
And also give a little bit the surrounding impressions of what we're doing in the i3D Bio project, if you would like that.
Perfect. That's great. We have a plan.
I'm really grateful. Anybody want to be a duo with Tom? Someone who has a challenge? Ha ha ha. We all have challenges. All right. All
I
right.
also would have an additional question, if that's still in time, about the sample
Please.
tracking system. And that is, I was wondering how much of that would you think is, so to say, easy to reproduce and implement at other sites? I'm always wondering how much, if you have a system that works in your place, that's super great, and then others are also having their systems in their place. And I was wondering what could be, so to say, the central positions where things go together. If, let's say, everything is working together with ELAB for the World as an ELN, and everything can communicate with that, and then that can communicate with Omero, what would be, so to say, the crosswalks and the crossroads that one could agree on somehow?
So I think the system is recomposed of 3 components that are largely interchangeable. And I think the biggest amount of effort was to figure out how to connect them. So I have something that collects the data, something that handles them, and something that delivers them to physical objects. And then those objects are scanned or recorded in a computer format. I think that you can essentially take those concepts and apply them. And of course, if anybody wants anything to to siphon off of us, we're very happy to provide you with the code and things like that. The problem is that our implementation is not really created by people that work on data science a lot. So it is a hack job that essentially works for us. I think effort to implement it at other place as is, unless you're copying everything, right? And you bind to infinity and you copy every single thing, would be very difficult. But kind of the system itself is easy to transfer. The things that are small, like which label printer to buy and what kind of software to run it, those decisions that we've made early on, I think were pretty good. And that kind of know-how is also costing a lot of time to develop. And we would be more than happy to share it with the group. But what we're actually working on is kind of, Infinity is very limited in what information we can transfer into that data sheet. So we're trying to build an API that's going to get that from the database. But that has been a slow process that eat a lot of snacks along the way.
Okay. Yeah, I'm thinking of a team in NFD Alpha Bioimage where I'm not sure yet, but we might bring them in touch with you. I'll ask them. Yeah.
Ideally, we would like some kind of open source solution for that kind of tracking, because as far as I know, it does not exist.
Yeah. Thank you.
Sure.
Okay. Thank you so much we are a little bit over time, so we definitely have 1 speaker for next, and then we'll try to find maybe another speaker, and then in February we're already lined up. And so great. Thank you so much.
Thank you, everybody. That's very inspirational.
Bye-bye.
Bye-bye.